Good evening, students. Good evening, Aditi. Good evening, Sanna. Good evening, Kirtana. And good evening, Manan Vignesh. Yes. So I hope you have attended the previous session, right? Human health and diseases, part one, where we covered what is the basic definition of health and how to achieve good health. And we classified diseases based on whether they are genetic or acquired. Right? Do you remember? Do you guys have any doubts on that? Okay. So, moving on with diseases, I'm going to explain or I'm going to discuss few of the diseases that are caused by the listed disease causing agents. So, these are the disease causing agents as you see here. The first one is bacteria, viruses, fungi, protozoa, helminths, Ricketshia, Chlamydia, Mycoplasma, and Prions. And I hope you will be aware of what are bacteria, viruses, fungi, and some would also be aware of what are protozoa. And these things are very new to you, right? You agree with me? These things are very new to you. But for each and every agent, let us take an example and analyze how to study a disease. For example, if you take bacteria, what are bacteria? Bacteria, to define what are bacteria or microscopic organisms which have a cell wall on their outer protective layer, and they produce diseases. Some of the bacteria are useful bacteria and some of the bacteria are useless bacteria. I mean, like, they are harmful bacteria. They are pathogenic in nature. The term pathogen, just keep it noted. The term pathogen means any infectious agent. Okay, pathogen is a common term used to refer to any organism that causes disease. Am I clear? So bacteria, some of them are useful bacteria. Like some bacteria are residing in our small intestine and large intestine, and they help us in digestion. They are useful bacteria, and some of the bacteria are present in roots of the nodules, and they help in fixing nitrogen, atmospheric nitrogen. They help plants to acquire nitrogen. They are useful bacteria. Okay, some of the class of bacteria like typhoid bacteria, okay, are the one that causes pneumonia, okay, the one that causes whooping cough. All these are diseases that are caused by bacteria. These bacteria are classified under the term called pathogen. Okay, so now I want you to analyze how this bacteria uh, get into our body and how it is starting or developing the symptoms, and what are the symptoms associated with each and every disease, and how these symptoms are developed and how we can treat them, or if there is any prevention, what are the preventive measures. Okay, so let's make a table like this. Now I want you to do this activity. Just make a table like this. Okay, for the first one, for the bacterial infection, let's take 5 bacterial infection now. The first column would be serial number. Okay, and the second column would be the disease name, the name of the disease. Okay, the name of the disease. And the third one would be the microorganism or the bacteria name. Microorganism that causes the disease. Microorganism. And the fourth column is habitat. Where do they present? Where do they develop the disease? Okay, habitat. And fifth column is symptoms. What are the symptoms? Okay, and the final column could be diagnosis and treatment. ENT. How do we diagnose it and how do we treat it? Can you make a table like this? Yeah. Text me once you're done with the table, okay? So quickly draw this table and let's analyze 5 diseases under bacteria, and some from viruses, some from fungi, some from protozoa, and one or two from helminthes, Ricketshia, Chlamydia, Mycoplasma, and Prions. I'll explain each and everything. Okay? Text me once you're done with the table. Kirtana, Aditi, Sana, Vignesh, and Nityashri. Just quickly draw this table. Okay, now, so after you finish with the table, the first disease is typhoid. Okay, typhoid was once a deadly disease. Okay, even now it is a deadly disease because it is affecting the entire, body, and it is very difficult to treat. Okay, so typhoid is the first bacterial infection. Can anyone tell me which organism that causes typhoid? And I know it is a bacteria, but I want you to give me the genus and species name of that particular bacteria. Okay, for any microorganism, we will have two names. That's called binomial nomenclature. The first name is called genus, and the second name is called species. Okay, so can somebody give me the binomial name nomenclature of the bacteria that causes typhoid? Okay, very good, Vignesh, Aditi, Nityashri, and Kirtana. Yeah. So I just need the name of the bacteria that causes typhoid. Yes. Okay, I'll tell you if you're not sure about it. Typhoid is caused by a bacteria called Salmonella typhi. Okay, Salmonella typhi. And do remember that when you're writing the binomial nomenclature, the first name should be started with the capital letter because it is a genus name. Genus name should always start with capital letter, and the species name should always be started with a small letter, lowercase. Okay, so bacteria called Salmonella typhi that causes typhoid. Okay, one GK question. So where do you think this bacteria can reside inside our body? Where do you think this bacteria can live inside our body? Which part? That comes in the fourth column, habitat. Okay, typhoid bacteria Salmonella typhi resides inside intestine. Okay, it resides inside intestine. So since we know that it is residing inside intestine, okay, so what are the causes, or how by how which mode we can get this bacteria inside our body? How do they invade our body? Can anyone tell me in what way they can gain entry into our body? Okay, Kirtana, stomach is fine, but intestine is the specific habitat for this bacteria. Yes. Through contaminated food and water, we can get this bacteria invading our body. Okay. So what are the symptoms of typhoid? What are the symptoms of typhoid? What are the symptoms of typhoid? Yes. Okay. So the symptoms of, yes, Vignesh. So the symptoms of typhoid include high fever. High fever, when I say it is 39 degree to 40 degrees Celsius. Okay. The fever would be between 39 degree Celsius to 40 degrees Celsius. It would be high fever, weakness, yes, Kirtana, fever, weakness of the body, and then headache, loss of appetite, yes. So these are the symptoms of typhoid. Okay, very good. So what is the diagnostic method we have and what are the treatment process we have? Okay, diagnosis is done through a special test called Widal test. Okay, Widal test. Yes, stomach pain, abdominal pain. Yes, they do have stomach pain. Yes, weight loss. Yes, weight loss is because of the loss of appetite. Vomiting is a rare case, but yes, vomiting can also be included under symptoms. Yes. So, Widal test. So, we need to study antibiotics. Vignesh, antibiotics is can be used for treatment. Now we are discussing about diagnosis. How do we diagnose this disease? How do we know that any individual is infected with this bacteria, Salmonella typhi? Okay. For diagnosing this disease, we have a special test called Widal test. We do this Widal test to confirm whether an individual is having Salmonella typhi infection in a body or not. Okay, we will confirm the presence of this bacteria through this Widal test. Okay, I'll explain what is this Widal test. So let me go to a particular slide here. So as I said, typhoid is caused by a bacteria called Salmonella typhi. But Salmonella typhi has got a new variety or it has got another species. It's called Salmonella paratyphi. Okay. There is another species that can also produce similar symptoms and the same disease. Okay. Salmonella paratyphi A and paratyphi B. There are two more strains that can cause the same disease. Okay, right? And because of this extreme weakness, and patients generally develop reddish spots or rose spots on their chest. Okay, it's just because their body and RBC has been affected, and because of that, they start this reddish spots. It's like patches of red colors all over the chest. Okay, so this is a fantastic story about typhoid Mary because she has been branded as super spreader. Okay, she has been branded as super spreader, and there's a story behind it. This Mary, this lady was was a chef in restaurants back in those days when she was working in different families. Back in those days, they used to work for families, right? She was working as a cook. Okay, when she was working for different families, she was the carrier of this typhoid bacteria, but without knowing that she was spreading the disease to different families. Almost like many families were infected with this typhoid disease, and they started developing the symptoms. Okay, and then the government took extensive steps to identify she was the carrier, and then she was taken to an island, and she has been isolated there. But she never supported the government. She just quickly escaped from that place, and then she started working for different families as cook again, without revealing that she carries the disease. She carried the disease. Okay, again she started infecting many families, And again, just like the second wave of corona, it was like second wave of typhoid. Many people were affected, many people were infected, and many were dead too. Okay, and then the government took serious note on this, and she was isolated until she was dead. Okay, so this is why she has been branded this name, super spreader, or she has been called Typhoid Mary. Okay, so now coming back to the diagnosis what we were discussing, it was done by a test called Vidal test. Okay, Vidal test is done in a slide. Okay, the slide usually has 6 circles. There is a broad slide. Okay, and you have 6 slides, 6 circles in this rectangular slide. Okay, what you do is that you collect the blood of the patient. Kirtana, yes, she did show symptoms, but she was not ready to accept that she was the carrier. Okay, she refused to cooperate with the government. Okay, so coming back to this Vidal test, we have 6 circles on this glass slide, and we have each and every circle marked with antigen. Okay, yeah. So they collected the serum of patients and they mixed the serum onto these antigens. Okay. This Salmonella typhi on the cell wall, if this is the bacteria, on the cell wall, they have something called antigen. Okay. Salmonella typhi had O antigen and H antigen. Salmonella paratyphi A and B had AH and BH antigen along with O. Okay, so this that is the reason why we do 3 tests in the same slide itself. This O and H, if it shows positive result, then it means Salmonella typhi is the causative agent. Okay, if it shows positive result on AH and O, then it means it is Salmonella paratyphi A. If it shows positive result on O and BH, okay, then it shows it is Salmonella paratyphi B. Okay, so what is this NC and PC is that? NC is negative control. Negative control is required and this PC is positive control. These two are required to judge whether we are giving proper result or not. We are getting proper result or not. For example, if I'm collecting the blood sample, serum sample from the patient, we know that he is carrying the disease. It is very sure that he is suffering from typhoid. Okay, in that case, if I'm collecting the serum or a serum from the patient, and if I mix it along with the antigen here, okay, I will get positive results in all these three, like this. Okay, we call this as agglutination. Agglutination meaning if you take the spoiled milk and if you start boiling it, you can see curdling of milk. Okay, the milk starts to curdle, it starts to group together, all the milk protein will start to group together, and all you get is the liquid separated from the solid. Okay, likewise this is called agglutination, like this as you see in the picture. Okay, all the antigens and antibodies, they come together and they just coagulate together, they group together. That is called agglutination. Okay, anyone has difficulty in understanding what is agglutination? If so, you can just raise your hand or you can just text me. I'll explain it again. Is this term agglutination clear to all of you? Okay. So once agglutination happens in positive control, obviously it would show the result. it would show agglutination, because the kit itself has a positive antibody or positive control. If you mix that with the antigen, then that will show the positive result. So this is to know how agglutination happens. Okay, so this will definitely show positive result. And wherever you get the positive result, wherever you get the agglutination, based on that, you will come to know what type of bacteria that causes the typhoid disease. Okay? Yeah, Kirtana. So this is the rapid slide test. This is the first level of Vidal test. Now we know that if agglutination happens, we know that the individual is carrying the disease. The individual is has been infected by the bacteria. Okay, he started developing the symptoms, and if he has not started with the symptoms also, soon he is going to be. Okay, and after that, how much he is carrying the disease. Okay, this is the qualitative test where we will come to know whether the patient or the individual is infected or not. Okay, but to know how much he has been infected, we need to do the second test called quantitative tube test. The first one is the rapid slide test. The second one is the quantitative tube test. Okay, it is very simple. Okay, you take 6 different tubes. Okay, 6 different tubes, and you take saline water. Okay, you take saline water. You take 1.9 ml of saline water in the first tube, and you mix zero point 0.1 ml, yeah, zero point 0.1 ml of the serum protein. Serum protein has been taken from the individual to be tested, or the patient who has been confirmed positive in the rapid slide test. Okay, this is the first qualitative test. Once Vidal test has been proved positive in the rapid slide test, for those patients only we will move with the quantitative tube test. Okay, so now in the first tube we are going to take 1.9 ml of saline water and 0.1 ml of the serum sample. Now it makes up to 2 ml, right? The total is 2 ml. In the subsequent tubes, you take 1 ml of saline water each. I'm going to take 1 ml of the saline water. Am I clear? Can you able to follow me? Okay, if something is disturbing or if you're not able to understand, just text me. I'll explain it again. So in the first tube, we are taking 1.9 ml of saline water and 0.1 ml of the serum sample. That makes it up to 2 ml. And in the subsequent tubes, we are going to take 1 ml of saline water each. Now, what I'm going to do is that I'm going to transfer 1 ml from first tube to the second tube. Okay, simply what I'm going to do is that I'm going to dilute the sample. Okay, so 1 ml I'm going to transfer it to the second tube. When I transfer 1 ml to the second tube, that makes it up to 2 ml. Okay, and after some time I'm going to take 1 ml from the second tube and I'm going to transfer 1 ml to the third tube. That makes it 2 ml in the third tube now. Okay, and again I'm going to repeat the same step until I don't see any agglutination. Okay, until I don't see any coloration. Okay. Okay, Kirtana, I'll definitely explain NC and PC again. NC is nothing but the negative control. Negative control meaning it is to show that there is no agglutination. Okay, this kit will have all the antigens and antibodies ready. Antigens, okay. Along with this slide, you will get O antigen, H antigen, H antigen, BH antigen, negative control and positive control. Okay, all these will come in small vials. Okay, now what we will do is that after collecting the sample from the patient, serum sample, I'm going to put the sample in OH, AH, BH, that's all. And I'm going to add O antigen to the first circle, H antigen to the second circle, AH antigen to the third circle, BH and antigen to the fourth circle. Okay. Now in the negative control, whatever has been given in the negative control tube, I'm going to add them. Most probably it would be distilled water and some of the agents, okay, some of the antigen. When I mix that, I'm not going to get any agglutination. And in positive control, I'm going to have agglutination, for which they would give you antigen and antibody. Any antigen and antibody that shows agglutination. Okay, so once you see the result, you can see agglutination in O as in the picture, and H and PC. And it also shows AH and BH, which means there is a combination of all the bacteria present. Okay, this patient, this slide shows that the patient is having all the strains: Salmonella typhi, Salmonella paratyphi A, and Salmonella paratyphi B. Okay, so this positive control shows agglutination, so you can compare and get the results, understand the results. Negative control, you don't see any agglutination. Okay, am I clear? Kitana, now did you understand what is NC and PC? Okay, if you don't understand still, just let me know. I will explain it again. Okay, okay, Vignesh. Okay, Kirtana, thank you so much. So now coming back to quantitative tube test. As I said, in the first tube, I have taken 1.9 ml of saline water and 0.1 ml of the sample, serum sample, that makes it up to 2 ml. And I'm going to transfer 1 ml to each subsequent tubes which contained 1 ml of saline water each. Okay. Now, as you see, the dilution has been increased by 20 times. Increased by 1 by 20th times. Like in the first one, it is 1 over 20. And in the second tube, the dilution factor has gone to 1 by 40, 1 by 80, 1 by 160, 1 by 320, and 1 by 640. And again, if the severity of the disease is more, you can start diluting it further until you get the negative result. Okay, you can keep on going it. Okay, you can take 8 test tubes and you can keep on increasing, or you can keep on diluting the sample until you get the negative result. Okay, this is how Widal test has been done, and this is how we know the severity of the disease also, how much the individual has been infected with Salmonella typhi. Am I clear? Okay. So now treatment process is very simple, because any bacterial disease can be treated with antibiotics. Okay. Antibiotics works in the fashion like it disrupts the cell wall synthesis. Most of the antibiotics, it stops the cell wall synthesis. Okay. Bacteria has been protected by the cell wall only. Okay, when the cell wall is not synthesized properly, when the cell wall is not fully formed, then it is susceptible to any physical damage. Bacteria, just imagine if our house is surrounded by grill gates and all that. Okay, if you don't put the lock properly on the grill gates, then you are susceptible to any physical damage. Your building is unsafe. Just like that, antibiotics stops the cell wall synthesis of bacteria. When bacterial cell wall synthesis is disrupted, its outer protective layer is disrupted. Okay, it is not properly functioning. When the outer protective layer is not properly functioning, this bacteria, the cell membrane and inner contents of the bacteria, is susceptible to any physical damage. That is exactly how antibiotics work. Okay, so just like that, the same principle applies for typhoid, because it is caused by a bacteria called Salmonella typhi, and simple antibiotic treatment is enough for typhoid. Okay, am I clear? Guys, am I clear? Any doubts? Okay, so let's go back to the list of pathogens that we were studying. So the first bacterial infection that we studied is typhoid. Okay, so just like how we have studied one disease, you need to continue the same method to understand different diseases. Okay, I'll give you some of the examples for bacterial diseases, and I want you to do the home activity to find what are the symptoms, how do we diagnose the disease, and how do we treat the disease. Okay, just note down the diseases. Bacterial disease, typhoid, we have done, and the second one is whooping cough. Okay, you need to find the causative agent as well. Okay, whooping cough, whooping cough. Third one is pneumonia. Okay, pneumonia. Pneumonia is as similar to corona what we are finding, prevailing now. Pneumonia is a general term that refers to any respiratory disease. Okay, the one that causes breathing difficulty. Okay, pneumonia can also be caused by bacteria, and just take it as a challenge and find the positive agent. Which bacteria causes pneumonia? Okay, and how do we treat it? And what are the diagnostic methods we follow? This is for bacteria. Just take three. Okay, three for the bacteria. And now coming to viruses. Can anyone tell me what are viruses? How do they differ from bacteria? What is the special feature about virus? What are viruses? What are viruses? What are viruses? Okay. So how do you differentiate between bacteria and viruses? Okay, Sana says viruses have their own genetic material. Of course, every living organism has their own genetic material. Okay, but viruses, viruses have only genetic material and the protein code. And a protein code. Their structure is very, very, very simple. Okay, every virus, if you take, they have their own genetic material. Either it could be DNA or RNA. Okay, depending on that, we have different classes of viruses. But still, every virus has their own genetic material and the protein code. This protein code has all the spikes, just like how you see in the picture here. Okay, it has spikes. These spikes are the receptors or the the protein material that helps in communicating with the host cells. Okay, the best factor about or the best fact about virus is that viruses are not classified as living nor non-living. They cannot be classified under living. They cannot be classified under non-living also. Okay. Yes, as Jay Rahul mentioned, they activate inside the host. Only when viruses are inside the host, they are alive. Outside the host, they are as same as dead. Okay. They are just particles. Only when they enter into the host and they just attach with the living cell with these spike proteins, only then they get activated and they behave like a living thing. Okay, as Keertana said, viruses are smaller than bacteria. They are alive only inside the host. Yes, Keertana, you're 100% right. And Aditi says microscopic infectious agent, which is a common or a broad term to define pathogen. Okay. Yes. So these viruses have genetic material and protein code. And I would tell you about one common disease. Okay. The first disease is common cold. Okay. Common cold. You may think that common cold is very simple. Like if I get a cold, okay, if I get cold, I may get cured without taking any antibiotics or any medicines in just 7 days. Just like how we watch in movies and we get a comic dialogue. If you take medicine, it will get cured in 7 days. If you don't take medicine, it will be cured in a week. Okay, both are same. Okay, common cold or any viral infections, it can be treated only by our immunity. Okay, any viral infection, it cannot be treated directly by antibiotics, just like how we treat bacterial infection or fungal infection. Viruses are very, very, very smart enough because they do not live outside the cell. They live inside the cell of the host. Okay, they use the machinery of the living cell, our own body cell. They take the advantage of using our own cell and they start multiplying in numbers. Okay, so we cannot destroy our own cell, right? So we are only dependent on our immune system. That's when any viral infection, we need to rely on increasing the immunity, improving the immunity. Only our own immune system can fight against any virus. Okay, but there are still antiviral drugs available, but they they are, I wouldn't say they are ineffective, but they can help us in improving our immunity so we can get enough immunity to fight against the virus. Okay, now coming back to common cold. Common cold is caused by a group of virus called rhinovirus. These are RNA viruses. Okay, rhinovirus. Okay, now we have a big threat in front of us, which is coronavirus. Okay, this is not a common cold, this is COVID, which is again a viral infection. Okay, third infection could be polio, poliovirus. Okay, poliovirus. So what do you know about poliovirus? Poliomyelitis was one of the biggest threats in early 80s and 90s and all that, because many children were having bone abnormalities and they were infected with poliovirus, and that's why government took enough measures to control it. Okay, government is taking enough measures to eradicate each and every disease, just like how we eradicated smallpox. Okay, we have smallpox vaccine. Okay, we are purely focusing on vaccine to improve our immunity to fight against any virus. Okay, so polio is caused by a virus called poliovirus. The disease is called poliomyelitis. Okay, just like how we did the table for bacteria, try to prepare a table for virus also. Write the common cold and it is caused by rhinovirus. What are the symptoms and what are the diagnostic methods we have and treatment. Likewise, you do it for COVID-19 and poliovirus also. Okay. One interesting fact about viruses is that when you start taking drugs to control virus, it gets mutated every now and then. Okay, you know what? There is no cure for common cold. There is no medication available for common cold. There is no one drug to control common cold. Okay, though it is not a deadly disease, it pushes our immune system down. So when the immune system is compromised, obviously it calls for other infections also. Okay, are you all aware about AIDS? It's a sexually transmitted disease. You'd have studied in grade 10 also, right? AIDS. What is AIDS? What is the full form of AIDS? What is the full form of AIDS? Anyone? Kirtana, Aditi, Rahul, Sana, Vignesh. What is the full form of AIDS? Yes, Vignesh, you can give me the full form of AIDS. Yeah, it is STD, sexually transmitted disease. I agree. But what is the full form of AIDS? It's called acquired immunodeficiency syndrome. The reason why it is called syndrome, any idea why it is called syndrome? Why is it called syndrome? Okay, Rahul, you're right. Acquired immunodeficiency syndrome. Yes. Why is it called syndrome? What's the meaning of syndrome? Syndrome means this is not just one disease that could kill the person. Okay. If any individual is affected by HIV virus, human immunodeficiency virus. Okay. If any individual is affected by only HIV, it is not going to die. He can live with HIV. But what happens in AIDS condition is that since HIV is damaging all your immune system, all immune cells, especially T cells, okay, the cytotoxic T cells. These T cells are the first line warriors for our immune system. When these are destructed, when these are damaged, there is no frontline immune system for us. So even a common cold, even a small infection can kill us. Okay, syndrome is a generic term that refers to the immunocompromised condition which can call for all other infections. Okay, that is what is called syndrome. Yes, Aditi, Vignesh, Kirtana, thank you so much for your answers. Syndrome means the condition where the individual has lost its, yes, lost its immunity to the core where he can invite all other simple infections also. Okay. Yes, that is syndrome. So you can just find what are the symptoms and what are the ways, modes of transmission and all that for these four viral infections. So now moving on to fungi. Have you heard of any fungi infections? Fungal infections. Generally, fungal infections are superior. Superior in the sense like they're all superficial. Skin diseases, okay, athlete's foot, okay, skin problems, trichoderma, the dermatitis, okay, all these dandruff, basically dandruff, all these are fungal infections. Okay, fungal infections. So now, with this corona situation, we have mucormycosis, right? The black fungal infection, which is one of the example for fungi. Try to find it. Try to find which fungal species is responsible. Yes, Rahul. Which fungal species is responsible for developing this disease, and what are the symptoms? You get black patches all over your nose and face here. Okay, black patches here. And if you look at the nerves inside the white aqueous humor, okay, all these nerves would be reddened. It will look like blood is oozing out of it. Okay, the entire eye would become red. Okay, generally fungi causes skin infection. So now, as a home activity, take the black fungal infection as an example for fungi and try to find the causative agent, symptoms, and diagnosis method and treatment. Okay, and yeah, next one is protozoa. What do you know about protozoa? Yes, Vignesh. especially those people who have been recovered from COVID, they also develop this. The reason why they develop this black fungal disease is that black fungal infection is that when we start using the mask, the one that we use to cover our mouth and nose, if we keep on using the same mask again and again, the moisture inside will invite this fungus. This is just the mold that you find on top of the bread. When bread is getting rotten, you get this black mold, right? The same fungus is responsible for this black fungal disease. But it causes severity in COVID patients, even though they have recovered from COVID infection, just because their immunity is much, much, much compromised. Okay, because our immune system would have been so tired fighting against COVID virus, and that's why this fungus can easily cause its infection in a much severe manner. Okay, yeah. So coming back to protozoa, what are the protozoan disease you have come across? What are the protozoan disease you have come across? Any idea? Do you know malaria? Do you remember malaria? Studying about malaria. The best example for protozoa is malaria. Okay. So malaria is caused by which protozoa? What is protozoa in the first place? Yes, Vignesh, it is not curable because it affects the main nerve that connects eye and brain. Yes, yes, Sana, malaria is caused by protozoa. And what is the name of that protozoa? That's what I'm asking. It is Plasmodium. There are three strains of protozoa that causes malaria. One is Plasmodium. Okay, Leishmania. Right, right, Aditi. Plasmodium vivax. That is one strain. Plasmodium falciparum. That is another strain that causes malaria. And there is one more strain called falciparum malaria. Yeah. Third strain is Plasmodium malaria. Should be written with lowercase. Yes, Sanna. Yes, Kirtana. It is caused by Plasmodium strains. These are the three strains that causes malaria. Do you know malaria is a disease that needs a vector, that needs the help of a vector? Okay, it is transmitted between two hosts. It has two hosts. Okay, the first host is mosquito and the second host is human. Okay, it is this mosquito acts like a vector and human is the final host where the disease is getting severe. Okay, if you look at the two stages of this plasmodium infection, let's take how it infects inside a human. Okay, okay. Now, if you take human, the sporozoites or the naive plasmodium strain, or the the newborn plasmodium strain. Okay, yeah. When sporozoites enter our blood system, when it enters our blood system, it enters into RBC. Okay, it enters into RBC and they start multiplying asexually. They reproduces asexually. When they reproduce asexually, they produce a chemical called hemozoin. Hemozoin. Okay, This is a toxic chemical. Hemozoin is a toxic chemical produced by this Plasmodium strain inside our human body. Okay, when this hemozoin is produced, this toxic substance immediately increases our body temperature and we get shivering. That's why, malaria is characterized by two major symptoms. One is the high fever and shivering. It happens periodically in every 48 to 72 hours. Okay, that periodicity is just because of interchange between the stages into host, human and mosquito. Okay, so for the asexual reproduction and for production of hemozoin, it takes 48 to 72 hours. And okay, and when this asexually reproduced gametophytes of Plasmodium vivax enters into mosquito, they start producing sexually. Okay, they start reproducing sexually and they produce sporozoites. So likewise, it is going in a life cycle. Okay, now in human, it is reproducing asexually, and this one enters into mosquito. In mosquito, it reproduces sexually, so that it produces viable plasmodium again. So it starts infecting another human wherever the mosquito goes and bites. Okay, this is how malaria happens. Malarial infection happens. Okay, the drug that is used for malaria is chloroquine. Chloroquine is the normal drug that is used to treat malaria. Since it's a protozoan disease, it can be treated with antibiotics. Am I clear? Any doubts? Okay, so up to this, I think you are a little familiar with the terms. And now coming back to helminthes. Who knows what is helminthes? Helminthes are nothing but, worms, a small worms that resides inside our body. Okay, it is again a parasite. It lives inside our human body and it starts developing the disease like the roundworm, ascariasis. Okay, ascariasis is the disease. Okay, ascariasis is the disease. It's caused by a parasite called Ascaris. It is just a worm. Okay, it is just a worm. Okay, we get this worm through contaminated food and water. Okay, once we get this worm inside, it starts utilizing our digestive system and it takes all the nutrients from our body and we feel bloated. We will lose, the feel of hunger. We will not eat properly. And since we are losing all the nutrients, we become anemic. Okay, internal bleeding. All these are symptoms of ascariasis. Internal bleeding, muscular pain, fever, anemia, blockage of intestinal passage, because more and more ascaris, it can just block the intestinal passage. How do we diagnose this? We take the stool test. Okay, since it is residing inside our small intestine, it comes out through anal opening. Okay, it comes out through anal opening when passing the stool. So we take a stool sample and we check for the presence of ascaris. If there is a presence of ascaris, then we confirm ascariasis disease and simply we take deworming medicine. Okay, any drug, deworming drug we can take. Okay, during early childhood, your mother would have given you in every 6 months deworming syrup. Okay, that would kill all these worms and you can just, get rid of this helminthus infection. Okay, any doubts? Next category is Ricketshia. Okay, Ricketshia. This is kind of very weird term because Ricketshia is not a new thing. It's just a class of bacteria. Okay, it is just a class of bacteria. But what is more special about Ricketshia is that Ricketshia, though being a bacteria, it can behave like a parasite. it can, reside only inside the host. Okay, it can live only inside a host. Like for example, a disease called Lyme disease. There is a disease called Lyme disease. This is caused by a Ricketshia species. Ricketshia species. Okay. So this Ricketshia species is present inside American dog tick. Okay. So have you seen ticks? A small, insect-like thing that sticks on the skin of the dog. Okay. So inside these ticks, this Rickettsia species will be present, and that produces Lyme disease in human. Okay. That produces Lyme disease. That is Ricketshia. Ricketshia is a class of bacteria that can live inside the host of a host just like a parasite. Okay. And next category is Chlamydia. Okay. Chlamydia. Chlamydia is basically a bacteria that causes sexually transmitted disease. Okay. Generally, sexually transmitted diseases are caused by viruses. But there is a class of bacteria called Chlamydia that causes sexually transmitted disease. But this can easily be treated through antibiotics because it is just another bacteria. So its cell wall synthesis can be disrupted by antibiotics, and simple antibiotic medication can cure this chlamydia. Okay. So the one that causes sexually transmitted disease is chlamydia, chlamydia trachomatis. This is the bacterial name, binomial nomenclature of the bacteria that causes the sexually transmitted disease. Chlamydia trachomatis. Any doubts? Okay. So the next class is Mycoplasma. Mycoplasma is yet another group of bacteria. But what is so special about Mycoplasma is that they don't have any cell wall. Okay, what we have understood right from the beginning of the class is that if there is any bacterial infection, antibiotic treatment itself is enough because bacteria can be killed by an antibiotic drug because it can disrupt the cell wall synthesis process. But Mycoplasma, since they naturally don't have any cell wall, they are very, very, very difficult to be killed by an antibiotic. Okay, so the mycoplasma infection they can be saprophytic or parasitic. The common infection is pneumonia. Okay, I'm just writing here pneumonia, and it is caused by a strain called Mycoplasma pneumoniae. Mycoplasma, mycoplasma. I know it is too much to remember all of them, but scientists have been named all of these in a simple logic. Okay, since it is pneumonia and since it is the mycoplasma that is causing the disease, the name of the name of the mycoplasma is Mycoplasma pneumoniae. They make it more simple like that. Okay, since the chlamydia is the one that causes Lyme disease, it is caused by Chlamydia trachomatis. Okay, try to remember, try to find the link between the disease, the class of organism, and the symptoms. Also, there would be some link between them, among them. Yeah. So the last category is prions. Okay. So far, anyone has any doubts? If you have any doubts, you can ask me. Otherwise, we will move on with prions. Any doubts, Kirtana? Vignesh, Sana, Rahul, Aditi? Any doubts? Okay. Okay, others? Okay, Vignesh. So the next category is prions. Prions is like I told you, viruses have only two parts. One is a genetic material, and the second one is the protein. These prions are smaller than viruses, where they don't have the genetic material. They have only protein. Prions are only protein, misfolded protein. Okay, or malfunctioning proteins. These proteins, only proteins, can induce our immune system. Okay, can induce our immune system. Prions are only proteins. How do they infect us is that when they enter into our body, our immune system is alerted. Okay, and these proteins will damage the brain proteins. The folding process, if this is the protein, this will undergo a three-dimensional folding. Okay, they will join with other protein sequences, and they will go folding and circling and all that, and these folding will be done in a wrong manner, misfolded manner, abnormal manner, and that will damage the brain protein, and that could be deadly. Okay, so some of them are these prions are majorly concerned or majorly related to brain proteins, and that is why it causes a disease. It causes diseases like dementia. Okay, dementia is gradual loss of memory. Okay, it causes diseases like dementia, and one classic disease is let me write it. Yeah, so it's Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease is a classic disease produced developed by a prion, and the major symptom is dementia, and obviously it will lead to death. Okay, so try to complete the table. Try to study all of these diseases in the table manner, like finding what is the organism that causes the disease, and what are the symptoms that are associated with the disease, and how do we diagnose it, and how do we treat it. This itself will clear all the disease causing agents and the diseases that we covered. Okay, so now the last topic for the day is that we'll come back to these questions at last. So last topic is Koch's postulates. Okay, Koch's postulates meaning there was a scientist called Robert Koch. He was a German microbiologist, and he was working on a anthrax disease in a sheep. Anthrax is produced by a bacteria. It is a disease that is caused by a bacteria. Okay, and he was the one who gave a proper definition for a term called pathogen, and he proposed few conditions. Okay, he came up with few conditions to classify anything under pathogen. Whether if you want to call anything as pathogen, they need to that organism needs to follow these conditions or satisfy these conditions. Okay, the first condition is that a diseased animal should regularly have that microorganism. Okay, if I'm suspecting that there is a disease prevailing in a community, and there is a death, there are some deaths due to a particular infection, the diseased animal or human should possess that pathogen. Otherwise you cannot possess that microorganism. Otherwise you cannot classify that microorganism under pathogen. That is the first condition. Second condition, the organism could be isolated and cultured on artificial or nutrient medium. Like if there is a dead person or dead animal, I'm trying to isolate the microorganism that was responsible for the death of the organism. Okay, that was responsible for the death of the organism. Now I have isolated it. Now I need to grow this organism in lab using artificial or nutrient medium. Okay, that should be possible, but there is an exception for this, because viruses cannot be grown or cultured on artificial medium. Viruses are exception for this. And there is a bacteria that causes leprosy. That bacteria will also, cannot be cultured on a nutrient medium or an artificial medium. Okay, there are two exceptions for this, but most of the other organisms or pathogens satisfy this condition. Third one, by inoculation of the culture, the same disease should appear in a healthy animal. Okay, if I'm culturing the organism in an artificial medium, if I'm taking a sample of it and inject into a healthy individual, the individual should, develop the same symptoms just like the dead one. Okay, that was the third condition. This is to prove that the same microorganism which has been taken from the dead is responsible for the disease. Am I clear? Okay. So the last one, the same microorganism should be recoverable from the body of inoculated animal or person. Again, it's just like a cycle. The same microorganism should be extracted from the infected person. We only infected the individual, but still the same microorganism should be present inside his or her body. Okay, these are the four conditions proposed by Robert Koch, Robert Koch, to identify any particular organism as pathogen. Okay, so this is about Koch's postulates. So yeah. So how do we expect some questions from this topic is that these are some of the sample questions, okay? So the first question, which of the following is a communicable disease? So now you need to remember what is the definition of a communicable disease. What is a communicable disease? It is transferred from one individual to the other directly, okay? Which of the following is a communicable disease here? The first one is TB. Okay, TB is caused by Mycobacterium tuberculei, okay, which is a bacterial infection. So that is caused by a bacteria, which is a communicable disease. Okay, diabetes mellitus is not a communicable disease. Okay, peptic ulcer is not it's a lifestyle dependent disease, and hypertension is again not a communicable disease. It is only TB. Okay, so you can expect some questions just like this. This is just a sample to show what kind of questions you can expect from this topic. So you need to know, just as I told you, the table in itself will give you every information about the diseases that we discovered, that we discussed. Okay. So whatever disease we have discussed, try to complete the table, and that itself is sufficient, more than sufficient for this topic. So for the next class, yes, for the next class, we will study immunity. Okay. What are the types of immunity and how do we fight against each and every infection? Okay. How does our body react to any infection? Okay. Yes. So students, if you have any questions, you can ask me right now or we can end the session. Yes, Nitya. Yes, Vignesh. Kirtana. You're right. It is TB. Yes. Guys, if you have any questions, you can ask me now. Okay, Kirtana. Kirtana says no doubts. So try to complete the table. Aditi says no doubts. Okay. What about Vignesh, Nityashree, Rahul, Sana? Okay, Vignesh. Thank you so much for the session and have a wonderful day. Thank you.